Okay, so having spoken a little bit about the general culture, suicide rates in Israel, and the complexities of the psychosocial findings, and we now come to the final presentation from Nadine, who will speak about some of the preliminary genetic findings. And just to remind you that really this whole exercise is to try and tease out the relationship between genetics, culture, psychosocial factors, psychiatric factors, as they operate in this very extended family who is overrepresented by suicidal behavior. And so the final piece now, Nadine will speak about the initial genetic findings.
Thank you. As Sami has mentioned, that although we've been speaking of suicidality for the whole symposium, in this kindred there are other high rates of psychopathologies, and mainly depression and anxiety disorders. So in addition to suicide, we have other psychiatric disorders that are elevated in this kindred. And in this presentation, I will be looking at presenting some initial results from candidate genes in relation to depression, anxiety, and suicide attempts. As I mentioned in the first presentation, that why we're studying the genetic etiology of suicidal behavior and the evidence from the family, twin, and adoption studies of a genetic role of genes in the etiology of suicide attempt. The same is true for depression and anxiety disorders. Family studies have shown the increased rates of depression in the first-degree relatives of depressed subjects as compared to the first-degree relatives of non-depressed subjects. And the odds ratio ranges between family studies from 2 to 5 times. There are also studies that have shown that parental depression increases the risk of depression in the offspring. So the offspring are at 3 times increased risk if one of their parents has been depressed. And also studies have shown that early onset of depression in the parent is also a risk factor for an early onset of depression in the offspring. And by early onset, meaning before age 13 in the offspring. And the increased risk is 14 times higher risk in this group. Similarly, the same is true for anxiety disorders. Family studies have shown that first-degree relatives of subjects with social phobia, generalized anxiety disorders, and agoraphobia, they are at higher risk of each of these disorders as compared to the first-degree relatives who have none of these disorders. And in the first presentation, I have shown you some of the evidence from our family study and looking at the offspring of probans who are concordant and discordant for suicide attempts. And that figure was based on cross-sectional analysis. More recently, we have looked at the offspring of attempters and compared them to the offspring of non-attempters in this study prospectively. So we have been following them up to 6 years, and consistently we find the same result, where the offspring of attempters are at higher risk as compared to the offspring of non-attempters, and they have an earlier onset of suicide attempt. The incidence rate ratio is almost 4 times. These results will be presented or will be published in the September issue, in this coming issue of the American Journal of Psychiatry, from this family study. Also, evidence comes from the adoption studies in depression and anxiety disorders, where as well the same is true. There is an 8-fold increased risk of depression in the biological relatives of adoptees as compared to the non-biological relatives. And as I mentioned previously, the increased risk is almost 15 times higher for suicide attempt. Twin studies have also provided estimates of heritability for the different disorders, and for depression that the estimates of heritability range from 37 to 43% in adults, and they were as high as 79% in younger children. For anxiety, the estimates of heritability for GAD is 49%, for agoraphobia 61%, and as I have mentioned in the first presentation, for suicidal behavior, the heritability estimates are 55%. The serotonergic system has been the target of the SSRIs and that are effective in the treatment of depression and anxiety disorders. And similarly, the serotonergic system is also the source and the focus of candidate gene studies for these disorders. So in this study, we have looked at 5 candidate genes in the serotonergic system that I will be presenting. The first one is the serotonin transporter, where a 44 base pair insertion deletion polymorphism in the promoter region of the serotonin transporter has been described. The short allele of this polymorphism is associated with a lower transcriptional efficiency as compared to the long allele. And the short allele has been found to be associated with depression, anxiety disorders, and neuroticism, which is more of a measure of emotional dysregulation that has been looked at as an intermediate phenotype for depression and anxiety disorders. Also, within the L allele of the 5-HTT polymorphism, there is an A to G functional variation, where the LG is more drives the expression of this drives the expression similar to the short allele. So the L allele, whenever there is a G, it acts like the short allele. In this study, in this kindred, the genotype frequencies for the 5-HTT are 16% are homozygous for the short allele, 48.4% are heterozygous, and 35.5% have the LL allele. And this is in Hardy-Weinberg equilibrium. The tryptophan hydroxylase is another candidate gene where TPH is an enzyme that catalyzes the conversion of tryptophan into 5-hydroxytryptophan, which is the very first and the rate-limiting step in the synthesis of serotonin. And TPH, the classical TPH, is expressed in the peripheral tissues, and it synthesizes serotonin in the peripheral tissues. But a differential expression that is called TPH2 has been described in the brain. So TPH then really does have an important role in the synthesis of serotonin in the brain, and that's the rationale behind it being an important candidate gene for depression and anxiety and suicidal behavior. In this study, we looked at a polymorphism, and 844GT polymorphism, which is in the promoter region of the TPH, that has been described and has been found in other studies to be associated with, to bias basically the reactivity of the amygdala, which is involved in emotion regulation. And the T allele was the one that is associated with greater reactivity of the amygdala. The TPH2 polymorphism, the genotype frequencies in this population are 42.3% GG, 46% TG, and 11.7% are homozygous for the T allele. And they are also in Hardy-Weinberg equilibrium. Other candidate genes that we have looked at and that have been suggested in the literature as important candidate genes for these studies are the serotonin receptors, where the 5-HTR1A1019 polymorphism, which is a polymorphism in the promoter region of the serotonin receptor, and the G allele has been found to be associated with suicide attempts in those who were exposed to a traumatic event or a stressful life event. And it has been found in other studies to be associated with panic attacks. The 5-HTR2A is also another polymorphism in the coding region, and it is associated with an amino acid change from histidine to tyrosine. And it does cause a blunted signal downstream of the receptor activation. And the fifth polymorphism is the 5-HTR2A, which is in the promoter region at position 1438, and the A allele has been associated with depression and seasonal affective disorders. In this study, we have looked at these 5 candidate genes, and we have conducted case control quasi-likelihood score test analysis, which have been developed by Bourgain and colleagues. And these types of tests are specific, and they have been implemented and developed for large inbred pedigrees such as our pedigree. And this test would provide basically 2 tests, the case control corrected chi-square and the quasi-likelihood score test for case control association. This is just a very tiny sample of the pedigree that shows the aggregation of the disorders. This is depression, anxiety disorder, suicide attempt, and this is suicide completion. And the pedigree, the actual pedigree, is basically like extremely long that really the room doesn't fit it lengthwise. So this is just a tiny section, and these are the consanguineous marriages between the parents of these individuals. In this study, if we look at the family history of these within the cases, the depression cases we have these analyses are based on 333 subjects for whom the data have been cleaned and analyzed, but we have another batch that are still in the process. So we have so far 100 cases of depression who have MDD, who met criteria for major depression disorder, and these are 71% of these subjects are the relatives they have first degree relative with an MDD, versus 12.9% who are second degree relatives, and as we can see, the rates drop in terms of the rates of MDD in the third and the fourth degree relatives. And very few have no family history, and these are basically the spouses that are inbred from the different families. So this is very typical of a disorder with a genetic etiology where the rates, the family history in the first degree relatives is higher, which is consistent with the studies that I have presented. Also, the same is true for anxiety, where 69% of these have a first degree relative with an anxiety disorder versus 10 to 11% with second and third degree relatives, and it drops for the third and no family history. Similarly, the same is true for suicide, where 55% of the suicide attempters have a first degree relative that has a suicide attempt. Looking at the relationship of the 5 candidate genes with depression, and the two tests that are provided, the QLS, the quasi-likelihood test, is a more conservative test as compared to the corrected chi-square. So we find a significant association between the 5-HTT and depression, and this one takes into account the functional variation. So the LG is considered to act as the short allele in this analysis. However, it's interesting that the short allele, the frequency of the short allele is lower in the cases as compared to the controls. And this is not consistent with studies that have been found in European samples, where the S allele is associated with depression in European samples. However, there has been some reports or some findings in Asian samples, in pharmacogenetic studies in Asian samples, where the short allele was associated with better response to SSRIs. For the TPH2, we have a 0.03 significance, and this one, once we correct for multiple comparisons, it doesn't survive that correction, whereas this one survives the multiple correction. So there is a trend, we can say, given the 0.03, where the T allele is really protective and the controls are protective for depression. And no significant association was found with the other candidate genes. For anxiety, the same is true with the 5-HTT, and this is significant even after correcting for multiple comparisons. Here the alpha is 0.01, given that we're looking at 5 candidate genes. So also the S allele is associated with a lower frequency in the cases as compared to the controls, which is consistent with the depression in this study. There is a trend for the TPH2, but it's not even close to significance. And here I missed to mention that the controls for the depression are those who have no depression, no anxiety, and no suicide attempt. The same is true for the controls in anxiety in this analysis. Cases have anxiety disorders, controls have no depression, no anxiety, and no suicidal behavior. For suicide attempt, we looked at it in 2 ways. So the first way is where the controls are subjects who have none of these phenotypes, suicidal behavior as well as depression and anxiety disorders. And the other way we looked at it is where the controls, they don't have suicidal behavior, but they could have depression or anxiety. And in the first analysis, although this is 0.038, but it's not statistically significant given our corrected alpha. And the same is true for the TPH2. There is here a trend that is not even close to significance. With the suicide attempt with the second phenotype, where controls are, they don't have suicidal behavior, but they could have depression or anxiety disorders. This one is close to significance. So, in conclusion, the 5-HTT is the candidate gene that we have found to be associated with depression and anxiety disorders in the study. However, the trend is opposite to what has been found in European samples, where the lower frequency of the S allele is associated with these disorders. And as I have mentioned, this is consistent with findings from the pharmacogenetic studies in Korean samples. And that's the importance of genetic studies, where we really need to examine different populations and with different ancestries and different environmental exposures, and study the effect of the environmental variation in the expression of these genes in order to really identify the relationship between the 5-HTT and these psychiatric disorders. Finally, I would like to specifically acknowledge 3 granting agencies: the Klingenstein Third Generation Foundation, NARSAD, and the National Institute of Mental Health, who have provided support of my training in statistical genetics, and they continue to do that. And also I would like to acknowledge all my colleagues for their great involvement, and really the study has been, and it's only the beginning, where we're now going to start the genetic linkage analysis, and special support to the families and the subjects who have agreed to participate in this study. Thank you.